Jianghan. I'm a third-year PhD student studying in the University of Bristol. And today my talk is microphysics of axial aerosol and in the airborne disease transmission context. My talk is a complementary talk to the last talk. Airborne transmission plays a significant role in the transmission of respiratory diseases such as COVID-19. So respiratory droplets can be generated upon different activities such as breathing, speaking, or coughing. Upon exhalation, during exhalation, multiple processes can occur due to the change in the gas phase conditions in the ambient compared to the humid lung. So first of all, we have the water evaporates because there is a lower ambient humidity compared to the humid lung. And secondly, during the water evaporates, possible phase change could occur in the droplet. And secondly, we have a CO2 evaporation due to the huge concentration gradient between the humid lung and the ambient, which cause the CO2 evaporate after being exhaled. This will cause a pH rise in the droplet. In order to study respiratory fluid droplets, a proper surrogate has to choose. Here I listed several recipes that has been used, which is artificial saliva and DMAM, which was introduced in the last talk, according to and compared them with the human saliva in terms of the chemical composition, the pH level. So artificial saliva from Wu et al. Study is a classic recipe that has been widely used in the respiratory experimental studies and also the modeling of respiratory aerosols. DMAM, Jubeco's modified Eagle medium, is a cell culture medium that has been used in the viral infectivity studies. And we can see that both of the surrogate systems share very similar inorganic ion species, including calcium, sodium, potassium, magnesium, bicarbonate, and phosphate. And they're all pretty much in a neutral pH level. And what's different is in the artificial saliva recipe, mucin is the only organic compounds, while in the DMAM, there are more glucose and amino acids, this kind of organics. Even we know what's in the respiratory fluid, the amount of ions can vary, highly variable among individuals, even within a day. So for example, when we are eating, the saliva state is called stimulated saliva, which has a higher flow rate with a higher ion concentrations. So in 1954, there is a study from Tian et al. Indicates the levels, the concentration range of the major ions in the respiratory fluid, which is showing in the gray bar in this graph. And also here I marked the DMAM and artificial saliva, the two surrogate systems that I used in this graph, which is showing in the orange and blue bars. And the other cell culture medium called MAM, which has also been used in our viral infectivity studies, are also marked. We can see from here that artificial saliva has the lowest concentration of sodium chloride compared to the other two, but has a higher potassium chloride level. And DMAM has the highest bicarbonate level compared to MAM and artificial saliva. And MAM has the highest sodium chloride concentration. These information will be useful to understand later results. So the experimental approach that I used to study the single aerosol dynamics is called comparative kinetic electrodynamic balance technique, which has been introduced by Rob. So here, what we typically measure is the size change and the size and phase change of droplet over time. So 4 phases will be characterized, which is homogeneous, inclusion, core shell, and crystal phases. Comparative kinetics, CK stands for comparative kinetics measurement, which referring to one a relative humidity probe dispenser that will dispense a well understood single component such as sodium chloride or water to track the real-time relative humidity. And a sample dispenser will dispense the solution that we are interested in. And one thing to mention is when the droplet is crystallized, the irregular light scattering pattern will not used to determine the particle size. And here is a typical graph that I can get from CKDB measurement, which is the drying kinetics at different relative humidity for different solutions. So on the left-hand side, we have a single component system, which is sodium chloride evaporation profile. Shows the radius change over time from 40% to 90% RH. And on the right side, we have the artificial saliva evaporation profile when we exclude mucin in the system. I will explain later. So essentially, the artificial saliva solution now is a mixture of inorganic salts. And above the efflorescence RH, the droplet will evaporate in the beginning because in the droplet there is higher water activity. When the water activity within the droplet is equilibrated with the ambient relative humidity, the droplet size will remain constant. And what's interesting is you can see the sodium chloride final size is much larger than the artificial saliva. This reflects the hygroscopicity of the two solutions. And below the, so we can see the sodium chloride is much more hygroscopic than the artificial saliva. And below efflorescence RH, the droplet will become crystals. So the phase change will occur. So it will form, for example, the non-spherical crystals, which is shown in the picture. So on the left is sodium chloride, on the right side is artificial saliva drying at 40% RH. And next we have the DMAM. Apart from the inorganic salts, DMAM has a large proportion of organics too. And this is how the DMAM drying at 40% RH look like. From these kinetic measurements, the equilibrium hygroscopicity profile can be estimated for different surrogate systems. And here I showed the hygroscopicity profile for different surrogate systems. On the left-hand side, we have the x-axis water activity versus the diameter growth factor. We can see there is a decrease in hygroscopicity from the MAM, DMAM and MAM sodium chloride, DMAM, and artificial saliva. We normally compare the hygroscopicity profile with sodium chloride because it's a well understood and it's a fairly hygroscopic solution. And because MAM has a large portion of sodium chloride in the ingredient, so you can see they are very similar to each other. While DMAM has more organics that reduce the overall hygroscopicity of the solution. And also we can convert the diameter growth factor to mass fraction of solute, showing on the right. This reflects the equilibrium chemical compositions in the droplet with varying RH. By measuring the hygroscopicity profile of a surrogate system, we are able to predict the evaporation behavior of this solution in a range of environmental conditions. And this has been examined by a newly published model called Single Aerosol Drying Kinetics and Trajectory Model, developed by my colleague Dan Hardy. you can see here, still the artificial saliva and DMAM evaporation profile. And the purple red dots are the modeled evaporation profile. They're in good agreement. So it's promising to accurately predict the evaporation kinetics through this model for more complicated solutions. And next, I would like to talk about the phase behavior of different systems. First of all, I explored mucin-free systems. So on the right is the artificial saliva without mucin, and compared with the sodium chloride, we can see they behave very similar. Above the efflorescence RH, the droplet remain homogeneous, while below the efflorescence RH, it forms crystals. However, in the DMAM, it behaves a bit differently because the presence of the organics causing much more complicated phase change, especially in the environmental humidity range, which is from 40% to 80% RH. And this is what we observed for MAM as well in our previous study. And the environmental humidity range also corresponding to the increased uncertainty of the viral survival in the aerosol phase. And now I'd like to talk about the influence of mucin on the droplet evaporation. So both of the graph is artificial mucin containing droplet compared with non-mucin droplet in two systems, which is artificial saliva and DMAM system at 40% RH, 20 degrees C. We can see here mucin does have an effect on the droplet drying. So it's slower down slightly about the evaporation rate for both of the systems. And there's slightly water residue in the droplet that makes the droplet stay in the trap longer, slightly longer. And also the mucin effect has been examined in 100 droplets in each of the system. And we can see that mucin delays the time to forming crystals and also increase the time to cause phase change in the droplet. And with this, I'd like to conclude and elaborate my future work. Water transport is crucial to understand the phase behavior and corresponding viral survival in respiratory aerosol. Mucin delayed the water transport and increased the phase change during aerosol drop evaporation, but only corresponding to minor effect of the early viral infectivity loss. There is a need to understand CO2 partitioning and its corresponding effects on pH change in respiratory droplets. Also, the need to explore the corresponding reduction in pH from condensable acidic species. Although the effect of pH on coronavirus stability have been explored in the bulk phase, the detrimental contribution of increased bioaerosol pH to airborne virus infectivity has only been recognized recently and requires further understanding. And with this, I'd like to thank my supervisor, Professor Jonathan Reed, and my collaborators and my research group, Bristol Aerosol Research Center. Thank you. I'd like to take questions now. Thank you.
Well, Jianghan, thanks for that. We've time for one or two questions if anybody has one. Yeah, well.
Hey, thanks for that. Nice talk. So you talked a lot about phase changes and the effect that might have on the viability of viruses and droplets. Do you have any idea of kind of what point during the phase change the infective, I don't know what infective potential or the how dangerous the virus would be changes? Is it a link between those things, or is it?
Yeah, this is a complicated question. I think you can kind of maybe we can think of the time scale. So there is an early infectivity loss and a longer time, like a minute, 10 minutes kind of time scale. I think during the process, the phase change plays a different role in terms of the different time scales. So for the shorter time, we see from Rob's talk that mucin helped the virus survival in the early stage, in the first few seconds, right? But in the longer term, I think, according to what we have done in the group, what we found is that the aerosol pH plays a significant role to the SARS-CoV-2 virus. But I'm not really sure about other type of virus. So yeah, it's a complicated question. Thank you.
Right, you can grab those if we've done it. Well, not actually grab those.
Thanks, very interesting talk, and I'm probably going to ask a very unfair question, but if within these droplets you have air particulates from air pollution, do you have any inkling about how that might change with the way they behave?
You mean in the droplet?
How that might change? Is that something that's looked at?
So particulate matter in what chemical region?
I think in just sort of air pollutants.
Oh, okay, yeah. So I think that's very important because the aerosol will interact with the ambient environment. So for example, what I mentioned earlier, the condensable acidic species, for example, I think the air pollution definitely has an impact on the ambient pH level. And that might affect the aerosols and also cause the corresponding effect to the virus. So, but I'm not sure I will. This is within my research scope, but it's something to think about. Thank you.
Okay, thanks a million, Jianghan. I think in the interest of time, we better move on.